Well, welcome to Click for Biology's discussion on the structure of lung tissue. Here we'll be looking at the various adaptations within the structures, particularly the gross structure levels, then the tissue levels and the cell specializations, all of which are addressing the surface area to volume issues that we raised in the previous video on gas exchange surfaces. Should you wish to, there's a PDF in the description which will allow you to make your own annotations as I go through this, and then you can back this up with a quiz, and indeed you'll there find links to other parts of the syllabus. So let's take a look at the structures of the lung, and we'll start with the gross structures as illustrated here. In the upper section of the respiratory system, we have the presence of the larynx, notable perhaps for the presence in there of a cartilage flap called the epiglottis. Now, the epiglottis, when one swallows, closes down and over the trachea, sealing it off, and in doing so, preventing food or water or drink and perhaps our saliva from going down into the tracheal system, the upper respiratory system. So the larynx then gives way to the trachea, the windpipe, and the windpipe is known to have cartilage rings, although these rings actually are C-shaped structures with the opening towards the spinal side. So at the front here, the open part of the ring would be on the other side of this image. The trachea then descends down and branches into the right and the left bronchi. The right bronchi is, generally speaking, wider and more vertical than the left side. And in fact, for those of you who will go on to do medicine, aspiration of the lungs is usually done down through the right-hand side of the lung tissue, I understand. However, the two bronchi both have cartilage as well, usually complete ring structures. And then these in turn branch into the so-called bronchiole. So we have a continuous branching pattern of the trachea to the two bronchi, and then into the bronchioles like this. And then these are going to keep branching smaller and smaller and smaller until we reach the dead ends and the alveoli gas exchange surface. The entire lung tissue is surrounded by what are called the pleural membranes. There's an inner and an outer membrane, and between the two is the pleural fluid. Other notable features of the large structure of the lungs is that on the right-hand side here, there are 3 lobes, and on the left-hand side, there are 2 lobes. The reduced lobes on the left-hand side, probably to accommodate the heart, which would be in this position, and the actual separation of the lung tissue into lobes is often associated with limiting the spread of infection. At the microscopic level, and in fact we're down to a diameter of 300 micrometers for the bronchial diameter, terminates in these structures called alveoli, in which we can see from the images this looping structure here suggests an increase in the surface area to volume ratio, a kind of folding pattern. So we have an increase in the surface area. Notice the proximity of the capillaries to the alveoli space, and this is implying short diffusion distances, which would therefore give us fast diffusion times for more efficient exchange of the oxygen and carbon dioxide. Now, the lining of the lung in this region here is known to be moist, and the function of this moisture within the lung is to improve the gas solubility of both carbon dioxide and oxygen. The moisture of the lung is derived from the plasma of blood passing through the lung. This image here was taken in our laboratory and illustrates just how delicate the lung tissue truly is. We note two different types of alveoli cell. The so-called pneumocyte type 1s, as shown here, are those which are the thin ones, which are reducing the diffusion distance for oxygen and carbon dioxide exchange. And then we have pneumocyte type 2 cells, which are responsible for the secretion of what are called surfactants. Now, surfactants, the function of which is to prevent the lung tissue from collapsing, and because of the moisture which is in this area here, there is the potential for hydrogen bonding across the surfaces and to make it difficult for the lung tissue to be inflated. And so it's kept apart by this so-called surfactant, which is in there. For scale purposes in the diagram, so that you can appreciate how delicate and how small this is, notice over here in the capillary, these red circles are actual individual erythrocytes or red blood cells. So this gives you an impression of just how delicate the lung tissue is and how difficult it is to actually create an effective drawing and diagram of lung structure. Let's take a look at cell adaptation. So the diffusion distances are not just small because of the size of the structures, but because of the intimate relationship between the capillaries and the pneumocyte ones. And I've tried to illustrate this here. So on this side we have the alveoli space, and indeed on this side we have alveoli space, and the red structure illustrates the capillary passing through the lung, and you can see the arrows indicating the movement of gases. But notice how the pneumocyte 1 is wrapped around the capillary structure, and therefore the diffusion distances are very, very small. And indeed they are reckoned to be in the order of 0.5 to 2 micrometers in distance. And so diffusion times are less than a quarter of a second. So those are some of the adaptations that we'll find at the growth structure, at the tissue structure, and at the cell structure, all designed to increase the surface area to volume ratio and to overcome diffusion distance issues. In the next video, we will consider the process of ventilation and thorax structure so that we can understand better how gradients, concentration gradients, are created and maintained.